Look at any living organism, and you will find that its complex physiological systems begin as a single, isolated compartment suspended in an aqueous environment. This compartment is the cell. While these units vary significantly in size and shape, they share a common structural blueprint that serves as the foundation for all body systems. This diagram illustrates the cell's physical boundary, the plasma membrane, constructed from two opposing layers of phospholipids. The spherical phosphate heads are hydrophilic, facing the aqueous environments outside and inside, while their hydrophobic fatty acid tails point inward, away from water. The bilayer also relies on supplementary lipids to function. Cholesterol molecules wedge between the fatty acid tails to regulate membrane fluidity, while thin carbohydrate chains called glycolipids extend from the outer surface to maintain structural stability. Dotted throughout this lipid structure are two distinct types of membrane proteins. Integral or transmembrane proteins fully span the entire bilayer from outside to inside. In contrast, peripheral proteins attach exclusively beneath the inner layer of hydrophilic heads. These embedded proteins execute four primary tasks. First, they act as specific receptors to capture external messages. Second, they form ion channels to safely transport charged particles across the boundary. Third, they function as adhesion molecules to lock cells together. And fourth, they act as localized enzymes to catalyze chemical reactions right at the boundary line. The plasma membrane is therefore an active, highly regulated biochemical interface. It strictly dictates what enters and exits, preventing chaotic interference from the surrounding environment. Moving into the interior of the cell, we reach the nucleus. This is the secure storage center for the genetic material and the site where DNA replication and gene expression occur. This diagram details the nuclear envelope, constructed from inner and outer concentric phospholipid bilayers separated by a gap. Protein complexes form nuclear pores to bridge this barrier, strictly controlling material passage. Deep inside, the nucleolus synthesizes ribosomes, which exit through these pores into the cytoplasm. The nucleus operates as an isolated vault, protecting the master genetic blueprints while safely exporting the physical instructions required for protein production. The nuclear envelope's outer layer continues into the endoplasmic reticulum. It folds outward, forming a massive network of connected, flattened sacs called cisternae. We identify the rough ER by the dense covering of ribosomes attached to its outer surface. This is the primary manufacturing site for both secretory and transmembrane proteins. As a polypeptide chain emerges from a ribosome, a specific amino acid sequence acts as an address tag, locking the complex onto the ER membrane. Transmembrane proteins embed into the membrane wall, while secretory proteins pass through the channel into the empty interior lumen. In contrast, the smooth ER lacks ribosomes. This section is dedicated to synthesizing lipids and storing calcium, which acts as a trigger for various cellular processes. Once synthesized, these newly produced lipids and proteins are packaged into transport vesicles that pinch off from the edges of the ER membrane. Vesicles travel to the Golgi apparatus, a stack of disconnected cisternae. Here, proteins undergo post-translational modifications, changing shape or gaining attachments as they shuttle between sacs. By the final sac, modified proteins exit in new vesicles, routed to the plasma membrane, lysosomes, or storage. The endomembrane system functions as an integrated factory line, translating genetic code into physically routed biomachinery. Managing the physical waste generated by this factory requires lysosomes. These specialized vesicles are packed with hydrolase enzymes designed to break down macromolecules into recyclable building units and to digest phagocytized bacteria. To prevent accidental damage to the cell, there is a biochemical failsafe. These destructive enzymes are only activated by the highly acidic environment maintained inside the lysosome itself. Supplying the power for all this activity falls to the mitochondria. This is where chemical energy is extracted from food compounds and stored in energy-rich molecules. This cross-section details the mitochondrion's dual membrane architecture. An outer boundary encompasses an inner membrane folded into deep ridges called cristae. Cellular respiration utilizes both spaces. The citric acid cycle occurs within the fluid matrix, while oxidative phosphorylation uses the folded cristae for energy extraction. By isolating extreme acidity and high-energy chemical reactions within their own specialized membranes, the cell protects the delicate cytosol from self-destruction. Suspending all these organelles is the cytoskeleton. This network of protein filaments provides structural scaffolding and establishes internal transit routes for the entire cell. The smallest filaments are made of the protein actin. These microfilaments enable muscle contraction, drive cellular movement, and support structures like microvilli. The middle tier is formed by intermediate filaments. Their protein composition varies by cell type, and their primary role is providing mechanical support. The largest tier consists of microtubules, hollow tubes built from 13 protofilaments of tubulin dimers. Their growth originates from the centrosome, an organizing center composed of two perpendicular centrioles acting as anchor points for the expanding microtubule web. This network holds organelles in place in non-dividing cells. During cell division, it reconfigures to form the mitotic spindle to guide chromosome movements. Microtubules also drive the motion of cilia and flagella. The cytoskeleton transforms what would be a loose liquid compartment into a highly ordered and architecturally sound machine. Zooming out, we see the fully assembled cell. Every distinct system we've observed operates simultaneously and interdependently. The eukaryotic cell is an actively regulated biochemical matrix where thousands of reactions occur side by side without interference. Specialized external structures, such as the microvilli in the small intestine or the flagella on a sperm cell, are macro-level expressions of these internal architectural systems. Every surface function relies on the precise extraction of energy, which powers a manufacturing pipeline, which in turn executes the instructions encoded in the master DNA blueprint. This hierarchy of internal form and specialized function is what allows a single compartment to operate with the complexity and precision of an industrial machine.